Hi, everyone. We're just giving another minute to the people who've registered and are joining us. All right. I see people are still joining. Okay, let's start. Kia ora koutou katoa. I'm Lana from Fulbright, New Zealand, and it is wonderful to have you join us for today's Good Works webinar. We have people joining us from across Aotearoa, New Zealand, the United States, and around the world, including Fulbright alumni and specialists working in biosecurity and environmental science. It is fantastic to see such a wide community gathered for this conversation. We have enabled Q&A, so if you've got a question for our speaker, just pop it into the chat, and we'll do our best to get to it at the end if we have time. For those who might be new to Fulbright, we are a part of a global exchange program that's been building connections between New Zealand and the US for nearly 80 years. Our mission is simple but powerful: to foster mutual understanding through education, research, and exchange. Every year, Fulbright supports talented New Zealanders and Americans to study, research, teach, and collaborate in each other's countries, building partnerships that last a lifetime. Now, Fulbright has become this amazing worldwide community, people who keep in touch, share ideas, and keep making an impact wherever they are. The Good Works webinar series is a way of showcasing the incredible work our alumni are doing in their communities, in research, and across disciplines. In today's Good Works webinar, we'll be exploring something most of us never think about: the atmosphere above us, and how it acts as an invisible highway carrying pests, spores, and pathogens across borders and oceans. Understanding that movement is vital for protecting our crops, our ecosystems, and our communities. Our speaker, Dr. David Schmale, is a pioneer in this field. He's a professor and director of the Translational Plant Sciences Center at Virginia Tech, and his groundbreaking research reveals how pathogens travel through the air, linking environments in a way that shape both agriculture and biosecurity. David is also a Fulbright US Scholar alum, having spent time in New Zealand in 2020, where he collaborated with the researchers at the University of Waikato to develop new remote sensing tools to detect and respond to harmful algal blooms. With $24 million in research funding, more than 120 publications, and lectures across 12 countries, his work is reshaping how we view farm security as national security. Please join me in giving a very warm welcome to Dr. David Schmale.
Thank you very much, Lane. I appreciate that. Let me go ahead and share my slides. Fantastic. Laney, just give me a quick little nod. Can you see the slides okay? Yes? Is that correct, Lena?
Yes,
okay,
yeah,
very
we
good.
can see the slides.
Okay, Very good. I can hear you now. Fantastic. Thank you. Always important to do a double check when we're running things virtually, make sure everything is running smoothly. Wonderful. So today I'm going to talk with everyone about unseen travelers, and there'll be quite a few stories from New Zealand and other places around the world as well, including, of course, our home here in Blacksburg, Virginia. And Lena and I were talking a little bit earlier about weather. We actually just got our first snowfall a couple days ago here in Blacksburg, Virginia. Of course, as I know, New Zealand is warming up and heading into warmer summer months. I'll start and I'll finish with a thank you. And of course, all of this work wouldn't be possible without just the wonderful lab group. That's my lab family. And of course, I'll start by thanking Fulbright. I'll be talking quite a bit about Fulbright and sharing a little bit of my experience that provides the ground for this presentation and the work that I'll be talking with you about today. Sponsors from the National Science Foundation, the United States Department of Agriculture, NASA, the Institute for Critical Technology and Applied Science, the Translational Plant Sciences Center that I direct, and also a See and Learn Foundation on Saba Island, which I'll talk about briefly today. So I think it's important to follow with a quote from the late Senator Fulbright. The essence of intercultural education is the acquisition of empathy, the ability to see the world as others see it, and to allow for the possibility that others may see something we have failed to see. And indeed, empathy in particular is something that is needed among our world leaders at this very moment in history. So I'd like to tell you a little bit about my Fulbright journey. On the 1st of January 2020, my family and I, we landed in Christchurch. We actually spent the first 4 weeks on the South Island, having a wonderful vacation before we moved north to, of course, the North Island. We purchased a van, drove around, had a wonderful holiday, and then the Fulbright actually started on the 1st of February 2020, and that's my oldest, who's now nearly 17 on the left, Nora, and my youngest, Vivian, who's 14 on the right. On the 1st of February 2020, of course, we gathered at Fulbright headquarters in Wellington, and pretty quickly thereafter, we were off to a marae where we spent the night. So one of my first experiences with the Fulbright family was, of course, spending the night in the marae and, of course, participating in a number of activities there. And that was a very special experience that I understand is something that's quite unique for Fulbrighters, certainly while they're in New Zealand. Of course, I got settled into my office at the University of Waikato, as Lena had mentioned, and my girls, of course, grabbed their uniforms and got started at school there in Tamahiri, which was where our home base was, right outside of Hamilton. And of course, shortly thereafter, in fact, the very next month, the Fulbright program ended around the world quite abruptly. There were some 800 Fulbrighters scattered around the world, and because of the pandemic, of course, the program was ended and ended early. We actually received instructions to return home to the United States. I decided to stay as well as another Fulbrighter and sort of ride out the pandemic in New Zealand with my family. And of course, we were part of what many Kiwis, of course, remember if you were there during this period of time. The former Prime Minister Jacinda Ardern, be strong, be kind, we will be okay, and her constant reference to the team of 5 million, of which we were part of during this period in history. Of course, we were listening to daily updates in the living room of our little flat, just like many of you were. Those afternoon updates from the former Director General of Health, Dr. Ashley Bloomfield. This will require all our efforts, and I strongly encourage all New Zealanders to play their part. And with all of you, of course, we watched the level system and the throttling of that level system as eventually we moved into a level system that allowed New Zealand to be COVID-free. And that was a pretty special time when we shot out the other side, was able to engage in some incredible research with some folks, including the Ion Lakes team, Brendan Hicks there on the right on the water, of course, in the waders, and really all around New Zealand with the Lakes 380 team, just an incredible group of folks. And I really was very fortunate to be able to work with them. This was actually a drone photo of some work out of the Chatham Islands, and I'll share a video for those of you that make it through to the end of the presentation. That'll be a little reward for those of you that haven't had a chance to visit. Of course, part of this also allowed deep engagement with communities, Maori communities in particular, lake trusts Here I'm working with Elder Mary as she's actually using a small portable device that determines chlorophyll and phycocyanin ratios and is a proxy for sort of water quality from a water sample that was actually collected from Lake Rotorua. And of course, Susie Wood with Lakes 380 did some incredible work as she continues to do with young student groups. And I was really fortunate to be a part of some of those activities as well, working with local communities and talking about the importance of course, water security and healthy water systems. And of course, our adventures in New Zealand, of course, were complemented with incredible whānau. This is Vicky Foden and David White and the kids. We had many wonderful adventures, including visits to their batch and their family's holiday home on the beach. And I was sharing with Lena earlier how it was, we had a Kiwi Christmas on the beach, which is something that folks here in Virginia don't get to experience in the snow in December. So pretty special. Well, the late Fred Rogers said, often when you think you're at the end of something, you're at the beginning of something else. And certainly that's going to be the story here today as my time in New Zealand transitioned, of course, to other things and other connections. And really, the focus of what I'm going to talk with everyone about today is connections, interfaces where systems actually touch. This is an image that was generated with Sora, an incredible image generation tool by OpenAI that's, of course, free to use, sort of exemplifying this theme. And I'd like to show a couple movies to talk a little bit more about these connections here. A little dandelion, of course, that's has wind basically causing these wonderful seeds, what look like little parachutes, to move through the atmosphere. The wind and the plant here, of course, being those interfaces. Or the beautiful kingfisher here, a bird, as it dives out of the air, it enters the water, it grabs a little fish. And of course, that's the easy part. Now it's got to turn around and get back out of the water and into the air. And so here we've got an air-water interface. And of course, an animal that is crossing that interface, that connection. And of course, flight is something that connects life. And one of the themes of my talk today, of course, will involve the atmosphere. And just a couple of weeks ago, I had the wonderful opportunity of spending a week on Saba Island, a very, very small island in the Caribbean Netherlands. And I've marked it here on the map on the left. Of course, you can see it's located east of Puerto Rico, just south of St. Martin. And in fact, it is home to the world's shortest commercial runway. And it is a very short runway, just a short flight, about 12 minutes from St. Martin. And it's an incredible marine sanctuary, a protected area, and it's very popular because of its wonderful and incredible diving. And while I was there, I had the opportunity to talk about the concept that flight connects life, because on the island, of course, they have the West Indian mahogany. And this is a beautiful tree that produces these fruits that, of course, yield seeds which have the ability to fly. These are helicopter seeds, formerly called samaras, and as they drop, they spin like a helicopter, which allows them to, of course, travel some distance from the parent tree. And so I had the opportunity to work with these students to exemplify this concept through a really neat exercise where they made their own helicopter seeds, actually out of paper templates and little paper clips. And you can see them doing that here as we were walking along this one road where many of these mahogany trees, of course, are situated. And in this movie here, and I hope the sound is going to come through on this. If not, what you can be sure is they were quite excited. These children are going to be throwing these seeds up into the air, and of course they spin like little helicopters, and they were quite delighted with this. And of course their teachers, I'm sure, were thanking me at the end of the day, as I'm sure these made their way all around the school grounds and even inside the classroom. Well, the atmosphere is teeming, of course, with invisible life, unseen agents, and today I'll be talking about some of those agents and their journey. And of course, because of the COVID-19 pandemic, we are able to appreciate terms like aerosol and aerosolization like we never had before. And so those terms have become part of our local vernacular, and of course have given many people the ability to appreciate that things like viruses can actually be transmitted through the atmosphere from one person to the next. And you might remember those rules that caused a formal distance separation between individuals to limit the spread of COVID. Well, pathogens are transported over long distances in the atmosphere too, and this is important, and this is something that'll be featured in my presentation here today. This is Asian soybean rust on the left. It's caused by a fungus, and it's a devastating threat to soybean production worldwide. Here on the right is Hurricane Ivan. Hurricane Ivan came across the tip of South America back in 2004 and actually picked up spores of the soybean rust fungus, carried them in the hurricane, and then deposited them into the United States. These spores, of course, came into our country in a single storm in a single night. And there they started spreading quite quickly, the fungus did, throughout the southeast. And this is actually a video of the hurricane as it picks up those spores at the tip of South America and then deposits them largely throughout the eastern portion of the United States. Um, of course, the hurricane made landfall in September, but it wasn't until 7 weeks later that the first report of soybean rust actually was made in a small little field in Baton Rouge, Louisiana. And so for 7 weeks, of course, this fungus was basically reproducing and steadily progressing throughout the range in which it was deposited. Of course, dust particles are also transported over long distances in the atmosphere. In the United States, we get dust from the Sahara that travels across the Atlantic Ocean and deposits into the eastern U.S., usually in about June and July of most years. The image on the right actually shows that dust crossing over the very same region in Sable Island where, of course, I highlighted the flying seeds. This is an image above Puerto Rico, actually in 2024 in July, on the 18th of July, and you can see these kind of golden skies. These dust particles can also carry pathogens with them, bacteria, fungi, and some of these can, of course, remain viable during this long distance transport event. Asian dust also arrives to the western part of the United States, but on a different time scale, usually earlier in the season, around April or May or so. Of course, insects can be transported over long distances in the atmosphere, and we had a really interesting event just this past September where a large swarm of this invasive spotted lanternfly, which is spreading quite rapidly throughout the eastern part of the U.S., actually showed up on weather radar. And so this was quite a big event. Here on the left, you can see this sort of map, this radar. This is a radar that was tuned to detect, of course, rain and snow. There wasn't any rain and snow. There was just this monstrous swarm of spotted lanternflies. These voracious eaters, as if you will, of course, were moving in critical mass. And shortly after this event, I actually ended up in Washington, D.C., which, of course, this is where this was near, at an event at our innovation campus. And there were literally hundreds of carcasses outside this building as we were walking along the sidewalks that were relics of this big swarm of these insects. Now, of course, when we think about things moving through the atmosphere, particularly those from outside of the U.S. Coming into the U.S., we, of course, call these atmospheric incursions. These can be pests or pathogens, and these events, whether they're via hurricane, a dust storm event, or some other mechanism, can be particularly troubling, and of course, they're very difficult to detect and monitor. Somewhat recently, this past summer, our Secretary of the United States Department of Agriculture, Secretary Rollins, launched a new National Farm Security Action Plan. And essentially, this action plan says that farm security is national security. And so the agenda now with the USDA has been very much aligned with this action plan, promoting agriculture and economic prosperity, defending the foundations of ag and food in the U.S., and strengthening domestic agricultural productivity. And so many of our efforts now, of course, are aligned with this action plan. And of course, funding and efforts in this arena, particularly when the federal government emerges from the shutdown, of course, are very much aligned with this action plan. So I mention this as an important national priority and certainly something that will come full circle as I talk about some of the wonderful work that my colleagues are doing in New Zealand. So studying life in the atmosphere actually requires teams of people that are also connected, in particular biologists and engineers. I'm an aerobiologist by training. I study life in the atmosphere. And I've had the great fortune of working with my wonderful colleague, Shane Ross, who's here on the left in the image, for almost 20 years. And we've been studying life in the atmosphere and the transport of that life. Back in 2015, we published an annual review that talked about the transport of pathogen spores across different scales, starting with, of course, a source where these pathogen spores would be released from a crop or from a residue. They're sent through the atmosphere. They're, of course, a removal via rain or washout. and of course, different factors that can influence their ability to stay alive during transit. And ultimately, the pathogen, of course, being deposited on hopefully a susceptible crop species. Hopefully, if you're the pathogen, you certainly aren't hoping that to be the case if you're the farmer or grower. Now, in this review, we conceptualize this in terms of scales that we basically, as with increasing size, starting with the scale of millimeters, that would be what we call the pathogen scale, into the farm scale, ultimately into the regional scale. The regional scale for us would be on the order of a state. We, of course, have state governments, and then we have the federal government within the United States, and that little star shows where we're located in Blacksburg, Virginia. And then, of course, to the continental scale, which involves the ability of one country to be connected with many other countries via the atmosphere in particular. And so today I'm going to share with you a story that takes us from a leaf, from a raindrop, from a spore, and we're going to scale that, of course, across the leaf into these large-scale movement patterns that could ultimately result in these atmospheric incursions. And this is just a conceptualized clip, again, generated with Sora by OpenAI, but of course, as a video. So I'll start with a story that involves the pathogen scale. And here we're talking about, of course, a fungus that's producing spores. And these spores are going to be transported, of course, initially small distances. And so I'll introduce you to a model system that we've been working with in my lab for a number of years. This is a leaf rust on wheat. And you can see the fungus is producing these incredible blisters. They're formerly called pustules, urodinia, and inside each of these little blisters, you can find hundreds of urodinia spores. These are reinfecting spores, and so when the spore actually leaves the leaf surface, it can reinfect the same plant or infect another plant within a cycle of about 6 to 8 days, provided that there's moisture involved. We've done some incredible work with some wonderful colleagues in engineering. On the right, Jonathan Barreco, he's staring at a little droplet generator. And of course, here at the top, there's a little needle. And from that needle, we can generate small little droplets and mimic rainfall. And we can target those onto leaf surfaces. Here on the left is Sonny Young at Cornell University. He's got his hand on a high-speed video camera that will enable us to record basically these connections, these interfaces between that raindrop and the leaf surface when they happen at almost 1 million frames per second. And so if you think about a standard little iPhone, your mobile phone that you have, if you put it in slow-mo mode, maybe you're capturing at about 200 frames per second. We're able to do a lot more. And of course, when we do that, we can observe some incredible things happening. This is a raindrop falling onto that leaf surface with those spores covered in those urodinia. And what you'll see is, of course, that these spores, they're hydrophobic. They stick to the outside of these droplets as part of what we call satellite droplets, and then can be dispersed, of course, via this rain splash. But what was somewhat surprising, and what you may not be noticing, is this vast quantity of dry dispersed spores that are actually being thrown up from the leaf surface as a result of these vortices that are produced during this impact. And this was a surprising discovery for us, particularly during rainfall. When you think about rainfall, you don't often think about dry dispersal. You think about wet dispersal, but the vast quantity of these spores were being moved in these microscopic vortices. And when we tracked the individual traces of these spores, they were actually forming these microscopic tornadoes. And we had a really nice paper that described this phenomenon where these microscopic tornadoes can then carry these spores over greater distances than if they were traveling in these splash droplets. Now, some of you, if you've had a cold beverage, of course, in the afternoon or the evening, have noticed that condensation droplets, of course, form in the outside of that cold beverage, particularly if you're sitting in a warm area, or perhaps as you move into the holiday season, having a cold one on the beach. Those condensation droplets can also form on plant surfaces, particularly in the sort of the dew cycle in the early morning hours. And those condensation droplets, when they form, they can actually collide. And when they collide, the energy of that collision can cause what's called jumping or jumping droplet condensation. And so we had done some work to examine whether or not these jumping droplets could actually carry spores away from leaf surfaces. And in this video, you'll see that phenomenon actually happening. So condensation growing on leaf surfaces, and then those droplets colliding, and the energy of that collision carrying these droplets, of course, into the atmosphere. And this is by a former graduate student, Dr. Nathan. You can see that these droplets, of course, spontaneously jumping and at quite a distance away from the surface. You'll see this a number of angles. They can also be iterative. Here's a droplet here. It's jumping. It's going to land and collide with another droplet, and this time it'll bounce even higher and then come back down. And of course, to observe these, you have to have them, of course, in the same plane. And so we got lucky with that one. This is a top-down view looking at the leaf surface. And of course, we're going to want you to pay close attention to an area over here on the the video. A droplet is going to shoot right out towards, of course, the camera. And we're going to zoom right in and you'll be able to see this. And so this again is a jumping droplet. It's jumping as a result of that collision. And you'll see that there are actually 3 Uridinius spores sitting there on the surface. And after those droplets collide and it jumps, those Uridinius spores are carried away from the leaf surface via this jumping droplet, this pathogen scale transport. So we've done some really neat work where we've taken and generated condensation on these infected leaf surfaces, as you can see here on the left. We've taken a fan where we can generate different wind speeds, and then we can look at the combined effects of course, jumping droplet condensation and wind. And then we can catch these droplets as basically they're jumping on these color-sensitive papers. And these color-sensitive papers, when they get wet, the droplets actually turn blue. And of course, when you take a look at them, you can see the droplets and you can see the spores that are enclosed or encapsulated within the droplets. And each of these little red spores that you see here is a Uridinius spore that was carried via those jumping droplets. Of course, that's the pathogen scale. And so as we move up to the next scale in our sort of scalar transport model, we're also interested in transport across farms, individual farms. And of course here, this will be exemplified with a fungus that we've done quite a bit of work on in my lab, a pathogen called Fusarium graminearum, which causes Fusarium head blight of wheat and barley. And of course, these diseased heads that you can see here, or spikes, have premature bleaching, and the grain that's inside these heads is loaded with mycotoxins. And these are dangerous secondary metabolites produced by fungi that, of course, are a significant threat to the health of domestic animals and humans that would be consuming that grain. Well, the fungus produces airborne ascospores that are fired out of these structures called perithecia, and these ascospores can then move over some distance in the atmosphere. And so one of my former graduate students, Dr. Presson, set up some experiments at a research farm near main campus here in Blacksburg, Virginia Tech's Kentland Farm, where we intentionally infected a crop field with a clonal isolate of the fungus. And so Dr. Preston is releasing residues here that are infested with the fungus, this clonal isolate, across a very large area. And then, of course, attempting to recover the clone at different distances away from the source downwind. And so on this figure here, we have the x-axis, which is distance away from the source. On the y-axis, the recovery rate of the released clone. And of course, the farther we get away from the released clone, the lower recovery of what we actually put in the field. We were able to actually look at our recovery rates and compare those, of course, to meteorological conditions from our source. And you can see how dynamic this plume is of Fusarium graminearum over time. And this allows you to also appreciate how complex this process of atmospheric transport is, even on a local farm scale. The wind is not always blowing in a single direction. The pathogen is not always releasing the same amount of spores at the same time every day. And so it's very, very complicated. And of course, environmental factors can contribute to all sorts of variability in terms of the source potential and, of course, where that plume is going to go. And of course, it's not just pathogens that can be transported over long distances, it's also pollen, and this can be a real issue, particularly if folks are growing genetically modified crops adjacent to, say, non-GMOs or organic producers. And so we had a really interesting study that was funded by the USDA BRAG program, where we genetically engineered switchgrass to produce orange fluorescent protein in pollen grains. We had a special site in the state of Tennessee that was permitted that allowed us to actually release these plants. It was in a big clear-cut area. And so here you can see the forest all around, which was clear-cut in this area where we're doing this experiment. And then we have 2 different blocks of plants. These plants are transgenic, of course, expressing orange fluorescent protein in the pollen grains. And then as that pollen's released, we attempt to, of course, capture it with different devices. And this video will show you just a little bit of our experimentation and some of the work that we do. There's the source. These are the panicles producing pollen. The switchgrass pollen, interestingly enough, is really only produced during a very short interval in the sort of early afternoons. We have to have a big team and lots of assets that we deploy to be able to trap the pollen. Assets that are, of course, placed both within inside the field. This is our mobile laboratory, and Hope Krzyzewski, a wonderful technician that helps out. The little H's are little, basically equipment pads that we have placed at strategic distances downwind from the source. A variety of different types of devices, some that are like vacuums that sample air at a known rate, other that are passive that just collect the wind as it sort of blows through. And then, of course, we have sophisticated meteorological instrumentation that allows us to monitor wind speed quite accurately, turbulence, gusts, and other things. And of course, just some drone imagery as well, which allows you to see the site. One of the big innovations for us has been impinging devices, which are like vacuums for air. They take air samples, they bubble them through a liquid. We also use high volume filter samplers as well to sample large volumes of air in a short amount of time. And then we have drone-based devices that we put on drones, which ultimately enable us to capture samples at different altitudes and different distances downwind from the source. This is one of our platforms here with that impinger on top. And of course, this is a drone video of a drone. And so this is showing just once again how you can have these mobile assets to be able to move. And you can see the bubbles there in the impinger on top of the device as it's sampling air, bubbling it through that liquid and trapping those pollen particles. And this just gives you an idea of our very special, unique, permitted site for us to be able to do that work. And then, of course, we always like to show the good landings like this one. We certainly don't like to talk about the unplanned landings, only the planned ones. And I'm an FAA certified Part 107 drone pilot. I conducted hundreds of missions across New Zealand while I was there with my family for a year and a half. And it's something that I really enjoy doing. I really enjoy getting out and doing the field work. At Virginia Tech's Kentland Farm, we have an incredible facility that was set up basically for drone research. We have a big airstrip that's dedicated to drones and, of course, an on-site lab. And we've been using this facility to actually develop techniques with colored smoke emitters, and you'll see those shortly. One of the challenges with the systems that we work with pathogens or with pollen or even dust is you can't actually see the particles, and so you don't know where they're going. These colored smoke emitters offer different colors and different pulses of smoke in a very short amount of time. So these emitters are quite inexpensive. They're about $20 US a piece, and they produce smoke for a little over a minute. Of course, you can do sequential smoke releases. You can do different colors of colored smoke emitters. So now you're seeing a blue one. And then, of course, you can mix them. Here's a red smoke emitter. The different colored smokes have different particle sizes, and so you can put different colored smokes together and then collect them, and you'll see an example of that at different heights. We have a little system where we can, of course, trigger the colored smoke emitters at different altitudes here on a tower, and then use the drone to collect those particles. The goal being to ultimately allow our robots to autonomously be able to detect these plumes and stay with them and be able to track towards a source. And so, and they also make for some pretty cool visualizations as the drones themselves are flying in these plumes. Of course, as the plume changes, one of the challenges if you have a ground-based device is that your device may not actually be in the plume, but with the drone can move around and you can ensure that you're actually sampling the plume as it evolves during the release event. And so this is something that's really, really important and will be exemplified in the next video that I show, because plumes are very dynamic. And even just work with what we've done at the farm and also in some of our dispersal experiments with pollen and pathogens, things just don't often move in a certain direction. I like to show these pictures because this illustrates what plumes look like under different wind conditions. And so on the top, you can see a high wind condition where the plume is very shallow, close to the ground and somewhat narrow, whereas a low wind condition where, of course, there's no wind and the plume is going to be moving mostly in a vertical orientation, particularly if you have turbulent mixing during about the late morning, middle, early afternoon. One of the things we've done with our pollen release experiments is we've implemented, of course, these colored smoke emitters. Here's our second year of experimentation. You can see the switchgrass plots are much larger than the earlier video that I showed. And you can see how the plume actually changes direction almost immediately. And so it was going across the field, and then all of a sudden it changes. And this is just an overhead drone shot. And so you can see how quickly that plume direction and intensity changes. And so this, of course, exemplifies the source, which is releasing pollen grains that we can't actually see, but we can collect. And so this gives us an important visualization tool to then be able to map back to our actual sampling devices to understand and validate the transport of the pollen as well. We're using some AI tools now, such as a really cool tool by Meta called Detectron 2, so that we can take these plumes and then we can actually use the imagery to basically map these plumes and determine concentrations. And so here you can see these plumes. The video seems to be sort of getting caught up here a little bit, but you get the idea. The smoke concentration is on the right, and we can actually extract that just from the images themselves when we do a calibration with these AI tools. So incredible tools for us to be able to advance sort of state-of-the-art and plume-based modeling. Well, finally, the biggest scale that I want to address, and this will ultimately link us back to New Zealand, is a scale which is one that we call the continental scale. And so now we're talking about really long distance transport across oceans, connecting countries, for example. And I want to take you back to that same time in history when, of course, we were in New Zealand. You might recall that we arrived on the 1st of January of 2020. Many of you probably recall the Australian brush fires, which were absolutely devastating, and they produced a ton of course, smoke that traveled actually across the ocean, and some of it made it to New Zealand. On the 2nd of January 2020, after we had landed in Christchurch, we'd purchased our car. We actually drove out to Akaroa, and this is an image of the peninsula right down, and you can see how hazy the sky is. And the sky is hazy because of all of the dust, of all of these smoke particles that came over from the Australian brush fires. And so my wife and my children and I, we all had itchy eyes. It was kind of hard for us to breathe. And it was because of course, the smoke that had traveled across the ocean, exemplifying sort of this continental scale transport. So this gives me an opportunity to introduce an incredible colleague, Dr. Ilse Pretorius. I think she's on this call. I did see her name pop up and want to highlight some Ilse's incredible work. She actually reached out to Shane Ross while I was still in New Zealand, just before I'd actually returned with my family to the US. And was really interested in developing a framework with Lagrangian coherent structures and modeling potential connections between New Zealand and Australia. And so I had the opportunity to actually visit Ilse at Scion, which is where she's based, and talk to her about this concept while I was still in New Zealand, and it ultimately led to a very fruitful collaboration. Ilse developed some of these LCS tools, which of course allowed us to model these pathways, which we called air bridges at the time, on the bottom that conceptually connect Australia and New Zealand. And then on the top, interestingly enough, these LCSs, of course, were correlated. These air bridges were correlated with satellite images of that smoke from those Australian brush fires. And so you can see that here, these modeled LCS air bridges, and then on the top panels, the actual satellite images of that smoke traveling across the ocean, of course, to New Zealand. I also went even further to look at these single bridges across different pressure surfaces during different times of the year. Here, of course, during November through April 2017 through 21, providing these different points of connection, of course, from Australia to New Zealand. And then yet another time of the year, May through October, during that same period, again, looking at these connecting points. And so this became a really compelling idea that Ilse and colleagues wanted to investigate in the concept of national security for New Zealand. Is there a way that the modeling of these LCSs and these air bridges could allow New Zealand to be aware of any potential pests or pathogens that could, of course, be part of incursions that could be moving from Australia to New Zealand? And so fortunately, Ilse and colleagues, including Tony Withers and many other wonderful folks, and I saw Steve Archer, he's another, saw his name pop up as well. I know he's here in the audience. Incredible colleagues really across New Zealand into Australia. And Shane and I, of course, are just a small part of this effort. We're certainly glad to be part of it. A big grant funded by MB to really study these pathogens and these insects that can be traveling across the ocean via these air bridges. And so just a really outstanding group of individuals. And Tony here, of course, by one of the moth traps, and you can see how excited she is as she's out there collecting these samples. I, of course, had the wonderful opportunity, along with Shane Ross, to come back to New Zealand last year in August of 2024. The Ilsa and the team had a kickoff meeting. It was wonderful to, of course, come back to New Zealand. I hadn't been back since we departed in June of 2021. My family and I actually spent a year and a half in New Zealand, and so it was a very special time for not only for us to be there, but for me to come back and spend this time with not only this group, but revisit some wonderful colleagues in Whānau on the North Island. I wanted to finish with another aspect of some of the work we've done. I've talked a lot about the atmosphere, but of course, the water is an important component of all this too, and connectedness of the water to the atmosphere and also land in this context is important. I'm going to show a video here that talks about some of our work really around the world in this context, starting here with some work that we did funded by the National Science Foundation out in the Atlantic Ocean, looking at the movement of rhodamine dye as a surrogate for oil. We released a large quantity of this dye, which is not a harmful dye, but it's a good way of sort of mimicking an oil spill. We sent assets out, such as this rib with a fluorometer to monitor concentrations of the dye. And then we've developed a 3D printed drone sampling platform to collect water samples. We dipped in and gathered samples of the dye. Of course, you can switch out the tubes. You can swap them out. That's me catching a drone out in the middle of the ship. We have underwater robots that we deploy as well that we're able to collect samples at different depths and then also monitor dye concentrations. We have a large robot called Big Bird that can deploy an underwater robot in an incredible location here, just minutes from our home, a local quarry where we can do some of this testing. Of course, you can do some underwater imagery as well to examine these dye tendrils, both in these controlled environments and then also in natural lakes, where we've had sensors on board these robots that allow us to do real-time monitoring. And here, a different dye, a fluorescein dye. We've also applied this to sort of search and rescue scenarios with mannequins here equipped with devices to understand how that they can drift and move in response to wind in particular as part of sort of search and rescue missions. Much of our work is also focused on harmful algal blooms caused mostly by toxic cyanobacteria. We've deployed our drone-based platforms to go out and sample these toxic cyanobacteria in the atmosphere and of course also in the water. And then get out and study these lakes. This is Grand Lake St. Mary's in Ohio. It provides water for about 3,000 people, and it's an extremely toxic lake with high levels of cyanotoxins. And it's not very deep, but nonetheless an important source of water for people that has to be cleaned up. This is Lake Erie, also in Ohio, that provides water for the city of Toledo, and we have the great fortune of doing some work there. We also have annual campaigns in Austria, in Europe. This is a beautiful glacial lake where we've done quite a bit of work sampling for ice-nucleating microorganisms that live in these glacial environments. And that's the Dachstein Glacier actually back behind there. And then just a little snippet of some of our incredible work out at Chatham Islands with, of course, the Lakes 380 crew. This is one of the Necklace Lakes, an incredible dune lake, of course, sandwiched with the ocean on both sides, and some of the drone imagery of the team out there at this lake. And so one of the sort of the overall themes of the talk, and I hope most of you in the audience have been able to appreciate, is really have a look where systems touch. Life moves in patterns that we're really only just beginning to see, and of course engenders more questions, more needs, and more effort. Observation is a first step towards stewardship, and this is really important, particularly when we're thinking about protecting our farms, our natural resources, and ultimately our people. And I'd like to argue that connection is actually the story of the planet. Humans, of course, have connections with one another, but we're also connected to all of this work, which is also connected to other things. And certainly I'd like to argue, and hopefully you've seen today, that the sky, the land, and the water are in a constant conversation. And I certainly hope that we all can listen just a little bit more. We certainly need to. I certainly like to thank you for your attention. And Lena, I think I've left us with some time, which is just about where we want it to be. at 4:45. While I take any questions that come along, I'll just share one final video and have it spool. Since I've done so much talking, I'm really interested to hear from all of you. This will be a video that just shows some of our campaigns out on the Chathams and some just incredible lakes, and you'll see that here. So Lena, I turn it back over to you to manage any questions, and I'll just let this video roll while we're going. And Lena, hopefully you're still there. Hello, Lena, are you there?
Thank you. Thank you.
Yes.
Thanks, David. That is fascinating. Your work really brings to life the connections between the air we breathe, the food we grow, and the ecosystems we depend on. Thank you. That is fascinating. Before we wrap up, I would like to share that our Good Works webinar series will be back next year. It is scheduled for February, and we'll have Diana Finch with us. And the webinar is titled Beyond the Numbers, Visualizing Data with Integrity. Diane will explore the art and ethics of data visualization using climate change as a backdrop, and she will show us how to interview data to uncover bias and communicate information truthfully in an age of misinformation. Right. So if you're interested in starting your Fulbright journey, we are suggesting that you visit our website, fulbright.org.nz. We have opportunities both for scholars and for graduates. I'll quickly check the Q&A box. Right. Don't have any questions. So in terms of trans-Tasman movements of insects and pathogens, is it routine, constant, or more associated with extreme events?
Can you repeat that question one more time, Lena? You were going in and out.
In terms of trans-Tasman movements of insects and pathogens, is it routine or more associated with extreme events?
Yeah, that's an interesting question. So that would certainly be something that would be better for Ilse and her team to answer, because they've been doing a lot of modeling sort of in this arena. I think the important part of that is Ilse's been able to monitor and predict some of these air bridges with some certainty. And when they do occur, and they're at a magnitude that would enable, of course, material to transfer across the Tasman Sea, then figuring out where to actually be to ultimately intercept those agents. And of course, Tony Withers and Steve Archer and others have to deploy sampling devices aimed at capturing the insects, the pathogen spores, among other things, when they're predicted to arrive. And that's a big challenge. You have to mobilize assets within a very short period of time, literally, within a window of about 48 to 72 hours, depending on the time of year. And then, of course, oftentimes the predictions, which are predicted to be a bridge, may not actually be a bridge. And so then, of course, the material doesn't exactly make it. So that's the best answer I have from that. Really, I don't know, Ilse, if she wants to chime in and answer any more, if she's still on the call, she certainly could fill any gaps if she's interested. Ilse, are you there? Maybe she's disappeared.
There's another question. Could you expand on aerolization processes, water, air interface, especially for cyanobacteria? Do you see uniform or selective aerosolization?
Yeah, that's an excellent question. So the question relating to aerosolization processes related to cyanobacteria in particular. So our focus has been on wind-induced aerosolization in particular. So wind sweeping across the surface of water, generating spume droplets, and understanding sort of how those droplets evolve both in size and of course time and space. There are other mechanisms of aerosolization, boats, of course, driving across a lake, waves crashing along a shoreline that can aerosolize toxic cyanobacteria. And so these processes are very complicated. They can occur, of course, out in the middle of a body of water. They can occur at interfaces with land, and they can be driven, of course, by human influences as well. Um, I didn't show a video of this in this talk, but we've done some work with a flume, which is like an aquarium in the laboratory, but very, very skinny, where we've used high-speed video to actually illustrate the droplets as they evolve at different wind speeds across the surface, and then how bacteria in particular can be sort of encapsulated in these droplets at different wind speeds, and of course, ultimately at different droplet sizes. And so we certainly have a lot to learn in this space. The state of Florida experiences regular red tide blooms, particularly along the Gulf Coast, that are caused by a dinoflagellate, Carenia brevis. And there are these toxins called brevetoxins, which can get aerosolized, of course, just with wave breaking. And so this is an issue usually actually late November, early December, almost every year, and really significantly threatens communities that live on or near the ocean, or that might be working, such as lifeguards, or even recreating, spending time, of course, on the beach. And so we need to better understand these processes, what drive, of course, the blooms themselves. And then as these blooms move to the surface of the lake, which is where they can become, of course, aerosolized, we need to understand these processes as well. So there's much to do in that context.
Wonderful. Thank you, David. We have another question. Hi, David. Great to see again what you were up to in New Zealand. A geeky comment inspired by your microscopic splashing experiments. Atmospheric pressure affects droplet splash dynamics. Reduced pressure does suppress the splashing effect. This is a complex relationship, but studied at these microscopic scales. I seem to remember that the reduction in pressure has to be as low as 50% of the standard pressure, so effects may be left at higher elevation, 5 kilometers in altitudes, and maybe not of any significance to liquid precipitation and vegetation due to extreme environments. But these physics may help in controlled closed chamber experiments if dynamics of spore ejection needs to be understood. Perhaps studied in your area or not important at all.
Well, there's a lot there. So that's really fascinating and very interesting. So in terms of sort of aerosolization processes or even droplet dynamics at different altitudes, certainly there's a lot of work to be done, in that space. I will say that we've been quite interested in the evolution of a raindrop from a cloud ultimately down to the surface of the earth, and the properties of that raindrop, the scavenging properties in particular, as the raindrops basically fall through oftentimes many kilometers of atmosphere, picking up particles and bringing them to the surface of the earth. The droplets can become bifurcated, they can split. Of course, splitting causes different sizes, multiple droplets. Of course, you have to think about the droplet itself also growing from a cloud condensation nucleus in the cloud. And of course, those droplets could be frozen, they could be hail, they could be snow. And so the dynamics are quite different. So we've thought a lot about this, and actually have conceived some experiments, but haven't been able to get them funded, where we drop high-speed video cameras off the side of bridges on flying rail systems to actually observe rain droplets as they fall through the atmosphere over tremendous distances. We certainly need a lot more work in this space. And then as we think about trees that could be sources of pathogens or even producing pollen at different altitudes in mountainous regions, how that pollen could be released and be thrust or brought down through valleys and into areas where they could cause huge sort of allergy problems for individuals that are particularly susceptible to pollen as a common allergen. And then, of course, main sources of spores being released. So a great set of questions. I don't know if I answered everything that was stated in there, but we certainly need a lot more controlled laboratory experiments. But most importantly, we need to do more work actually out in the field, deploying assets to be able to study these natural phenomena across much larger scales.
Thank you, David. We don't have any other questions, but people are very complimentary. Super interesting. Thanks for the great talk, David. Great talk, David, as always. And we also joining them at Fulbright New Zealand. Thank you, David, for your time, and thank you for building these connections between New Zealand and the US. We do appreciate it. And thank you for all who joined us today. Ngā mihi nui, and have a wonderful rest of your day or evening, wherever you are. Thanks so much, everyone.
Thank you.
See you.
Great to see you all again.
Bye.
Bye-bye.